Welcome to the Fungisphere webinar series, brought to you by the Sydney Infectious Disease Institute here at the University of Sydney. I'm Associate Professor Justin Beardsley, and it's my pleasure to introduce these talks, our passionate team, and share some of our mission with you. We're dedicated to increasing our understanding of all things fungal, be that human health, animal health, agriculture, ecology, or even the productive and industrial use of fungi. We are seeking to uncover the threats and the promises offered by these remarkable organisms. This webinar series is more than just a series of talks. It's a platform to bring experts in the field together to share their knowledge and hopefully to build new connections. It's a testament to the synergies that exist between researchers here, but also colleagues globally. Through these talks, we share some of the incredible work being done here in the University of Sydney, but also provide a platform for international experts to share their groundbreaking research too. So, whether you're a seasoned researcher, a student, or simply somebody with an interest in the fascinating world of fungi, you'll find something of interest in this webinar series. Together, we're going to explore the threats and the promises of the fungal world. Serisha Naicker joins us from the Centre for Healthcare-Associated Infections in South Africa. She's going to present her work on the virulence of cryptococcus. She's also going to discuss the challenges posed by the emergence of fluconazole resistance in clinical isolates over the last decade. Let's hear from Serisha.
Welcome to another session of Fungisphere, and for the guests which are not part of the Fungisphere this time, just a little background on Fungisphere. I think Fungisphere is a mycology interest group as part of the Sydney Infectious Disease Institute. It was generated like around 3 years ago as part of the Murray Bashir Institute for Biosecurity and Infectious Diseases, Emerging Infectious Diseases, and is headed by Professor Tanya Sorrell and Professor Ben Murray. We are trying to include as many mycology people interested within the university, but also from outside. And today, it's my pleasure to welcome Serisha Naicker. She is from the Institute for Communicable Diseases in Johannesburg, and she is there part of the Mycology Research Laboratory and Reference Laboratory, which is especially working together with Nalesh Governor, which many people probably know around us. So Serisha, she did her master's degree in science at the University of Witwatersrand in South Africa in 2012. And then she started to do an internship in mycology to be registered with the Health Professional Council of South Africa. And in 2013, she took up the position as a medical scientist at the Institute. When she came to Australia in 2015 to attend ISHM, she then decided to move on to do a PhD and partially taking this up as part of her work, where she works on cryptococcus, but because she did also work on Candida and other species, and Candida auris and Claprata, that she could only take out this position as a PhD student, as part-time student. So her PhD is focusing on cryptococcus and cryptococcosis in South Africa. And that's what she will give us an update today, because she just has about finished her PhD and is about to actually pass the examinations, and the thesis is in part went through, and she is co-supervised by Nalesh and myself. So it's a great pleasure, Serisha, to have her here, and we're looking forward to hear about the special aspect of cryptococcosis in South Africa, where we all work globally, and I think South Africa is a very interesting part of this. So please start to share your screen, and please give us your talk. Thank you very much.
Hi, everyone. Can you see my screen?
Yes. I think we have to go to a presentation mode, otherwise it's too small.
Is that better?
Yes, perfect.
Hi, so thank you so much, Wieland, for the opportunity and for the introduction. So today I'll be talking about the molecular epidemiology of Cryptococcus neoformans and Cryptococcus gattii species complexes in South Africa. The global burden of cryptococcal meningitis among people living with HIV/AIDS is about 220,000 cases per year, with approximately 180,000 deaths. 73% of global cases and 75% of deaths occur in sub-Saharan Africa. In South Africa, over 6,000 patients with laboratory-confirmed incident cryptococcal episodes were reported during 2019, with an incidence risk of 1 reported case per 1,000 HIV-infected persons. In South Africa, cryptococcus is the most common cause of meningitis. In South Africa, there were only 3% of viable isolates identified as Cryptococcus gattii during laboratory-based surveillance for cryptococcosis, and the rest of the isolates were identified as Cryptococcus neoformans. These are the reported isolations of C. neoformans and C. gattii across each continent. The pie charts show the distribution of clinical and environmental and veterinary isolates, and the bar chart shows the number of isolates that have been reported, with the number examined for molecular type shaded. And as you can see, there were very few African isolates that have been genotyped. In terms of the molecular type distribution across Southern Africa, for C. neoformans, VN1 and VNB are the most common molecular types. And for C. gattii, VG1 and VG4 are the most common genotypes. Other South African studies have been performed on sequencing Cryptococcus strains, but these studies have focused on patients enrolled into clinical trials or from clinical surveillance. So the aims of this study were to explore the genetic diversity of C. neoformans and C. gattii, to determine if patients' clinical characteristics are associated with cryptococcal molecular type, and also to determine the effect of molecular type on in-hospital outcome. We also evaluated the fluconazole susceptibility pattern of isolates from first episodes of cryptococcal disease. We also hypothesized that the VNB molecular type may be more virulent than the other molecular types, so we determined its virulence in a Galleria melanella larval model. And we also aimed to describe the population genetic structure of VNB and VG4. Since 2005, national laboratory-based surveillance for cryptococcosis has been conducted through the Germs SA surveillance platform. A case has been consistently defined as illness in a person diagnosed with cryptococcal disease at any South African lab based on any one of these following tests being positive during a 30-day period. This being a positive India Inc. test on CSF, or a positive antigen test on blood or CSF, or culture of crypto from any specimen. So the major advantage of our study was that we randomly selected a representative sample of clinical cryptococcus isolates from an isolate bank created through national population-based surveillance. This study design allowed us to explore the molecular diversity of the whole population of clinical cryptococcus strains from South Africa. So we randomly selected 251 C. neoformans isolates. We phenotypically characterized these isolates using Niger seed agar and CGB agar, and thereafter we genotyped these isolates using multilocus sequence typing. So this pie chart shows the molecular type distribution of 251 C. neoformans isolates. 82% were VN1, 10% were VN2, 6% were VNB, and 2% were VN4. The tree on the right-hand side shows the 67 different sequence types identified. We grouped the molecular types as VN1 versus non-VN1 to determine associations between clinical characteristics and genotype. Molecular type was defined as the dependent variable, and we included the following variables: sex, age, year, province, specimen type, HIV status, mental status, CD4 count, antiretroviral, antifungal, and TB treatment. We separately modeled the effect of molecular type on in-hospital mortality, adjusting for sex, age, mental status, CD4 count, antiretroviral, and antifungal treatment. So on our univariable logistic regression analysis, we found that males were more likely to be infected with a non-BN1 genotype. And compared to patients infected with a VN1 genotype, those with a non-VN1 genotype had a 50% reduced odds of dying after adjusting for sex, age, CD4 count, antiretroviral treatment, mental status, and antifungal treatment. Although the point estimate was 0.50, the 95% confidence interval spanned 1, and the p-value was large. We then performed fluconazole susceptibility testing by the CLSI broth microdilution method on 105 isolates of senior-formans, and we used published epidemiological cutoff values for interpreting the MIC values. So the average fluconazole MIC 50 and MIC 90 values for 105 senior-formans isolates was 1 microgram per mole and 4 micrograms per mole, respectively. Most isolates had a MIC value of less than or equal to 8 micrograms per mole, which is considered to be wild type according to published epidemiological cutoff values. There was, however, only one VN1 isolate with a MIC of 32 micrograms per mole. So we determined the virulence of the VNB molecular type by infecting Galleria melonella larvae with these 15 VNB strains and comparing this to a highly virulent H99 reference strain. So based on a previous South African study where patients infected with VNB had a significantly worse survival outcome, as seen in the graph by this yellow line, we hypothesized that this molecular type may be more virulent than the other molecular types. And then going through the results of the virulence studies, we found that the 15 VNB strains showed some heterogeneity in their virulence, as one strain killed as quickly as H99. So H99 is shown by the bright pink line, while most of the other strains killed more slowly. So we actually found that our strains were less virulent than H99 in this model. We also focused our whole genome sequencing on the 15 VNB isolates because this molecular type was known to be endemic to Southern Africa. So we used the Illumina MiSeq to sequence these isolates. We used FastQC for quality control, and we used BWA to align reads to the VNB reference, and we called SNPs by SAM tools. All of this was done in the NASP pipeline. So all 15 VNB isolates clustered in the VNB2 clade. The patients were distributed across South Africa as shown by the map. There were 9 isolates that clustered very closely together as shown by the cluster clade at the top of the tree. There were 4 other isolates that were scattered across the tree, and then there were 2 isolates that were closely related to an environmental Brazilian isolate. We then added 95 public samples, and we found that there were 6 strains that were closely related to our 9 strains in the cluster clade. So this tree just zooms in on those strains with less than 30 SNPs between these isolates. The cases did not cluster in time, as you can see, and we could speculate a common environmental source, but since the patients were scattered across different parts of South Africa, it's difficult to conclude how these strains could have been related. So our study confirmed that there's a high genetic diversity of South African senior-formans isolates. And due to the high genetic diversity of African clinical and environmental senior-formans isolates, it was hypothesized that this organism originated in Africa and then spread globally across to the rest of the world. This has been termed the out of Africa hypothesis. We found that there were 2 of our isolates closely related to Brazilian isolates, and recently genomic evidence has suggested that ancient migrations of the VNB molecular type has happened bidirectionally between Africa and South America. So moving on to the C. gattii study, we randomly selected 146 C. gattii isolates and we genotyped these isolates by MLST. The pie chart shows the molecular type distribution of these 146 C. gattii isolates. 70% were VG4, 27% were VG1, 2% were VG2, and 1% were VG3. And the tree just shows the 99 different sequence types that were identified. We also performed a similar analysis as the senior-formans study. So for C. gattii, we grouped the molecular type as VG4 versus non-VG4, with molecular type as the dependent variable and the various patients' clinical characteristics as the independent variables. We also separately modeled the effect of molecular type on in-hospital mortality. So on multivariable logistic regression analysis, we found that HIV seropositive patients had a 10 times increased odds of a VG4 infection compared to HIV seronegative patients after adjusting for geographical region, sex, and age. Although there were small case numbers, the confidence interval was very wide. On the multivariable analysis, patients infected with the VG4 molecular type had a 43% reduced odds of dying than those infected with other molecular types after adjusting for those variables seen on the slide. But the 95% confidence interval crossed 1 and the p-value again was large. We also performed fluconazole susceptibility testing by the CLSI broth microdilution on these 105 C. gattii isolates, and we used epidemiological cutoff values for interpreting the MICs. The MIC 50, the average fluconazole MIC 50 and MIC 90 values were 4 micrograms per mole and 16 micrograms per mole for 105 C. gattii isolates with a geometric mean of 4.53 micrograms per mole. Most of the VG1, VG2, and VG4 isolates had a MIC value of less than or equal to 8 micrograms per mole, which is considered to be wild type according to published epidemiological cutoff values. However, the 2 VG3 isolates, there were 6 VG1 isolates and 6 VG4 isolates that were considered to be non-wild type. We performed whole genome sequencing on 101 VG4 isolates. We concentrated on this genotype because it was the most dominant in our study. We found that there were 3 genomes that had a low quality, so we excluded them from the analysis. So we only analyzed 98 VG4 genomes. Again, we used the Illumina MySeq platform. We used FastQC for quality control. We aligned to a known VG4 reference using BWA and call SNPs by SAM tools in the NASP pipeline. So we then added 47 public VG4 samples into this analysis. There were many SNPs between isolates, but we observed 2 interesting clusters as indicated by these rectangles on the slide. This map just shows the 508 clinical cases in gray of cryptococcus gattii infection collected during 2005 to 2013. from 3 different South African provinces with cases that clustered closely together by genomic data they highlighted in yellow. So we then analyzed each of these clusters separately. The first cluster consisted of 10 isolates from the Gauteng and Limpopo provinces of South Africa. 6 patients were male and 4 were female. 7 patients were from Limpopo, 4 of which were diagnosed at the same facility, and 3 were from Gauteng. The cases, however, did not cluster in time. And then for the next cluster, there were 8 patients in this cluster. 6 patients were from Mpumalanga, from our study, and there were 2 isolates from a previous South African study. There were 5 male patients and 1 female patient, and 5 out of the 6 patients were diagnosed at the same facility. But again, the cases did not cluster in time. However, it does suggest a similar environmental source. So we observed a high genetic diversity with VG4 being the dominant molecular type in South Africa. This confirms studies undertaken in Zimbabwe, Botswana, and Malawi. VG4 has also been reported to cause infection in HIV seropositive patients, and we are unsure why this could be the case. But it is possible that various factors such as fungal population genetics and environmental distribution interact to cause infection in patients with varying immune competence. So since we found that there was one cluster of 6 strains isolated from patients living in one South African province, this made us speculate that VG4 may be widely distributed in the subtropical environment. So in a previous study done in Zambia, C. gattii was mostly associated with the central Miombo woodlands in Zambia, and this area is shown in red. So this area is dry and tropical. And the genomes of 6 isolates represented a new molecular type, VG5. This molecular type was found in hyrax-associated environments, making this a new environmental niche for C. gattii. We, however, did not find any VG5 isolates in our collection of clinical C. gattii strains. It is possible that this molecular type is strictly environmental and it's yet to cause clinical infection. However, further studies of clinical and environmental C. gattii strains from Southern Africa are needed so that we can confirm this and to further study the complex. And then moving on to the final part of the study, we compared the fluconazole MIC values between 2 surveillance periods, 2007 versus 2017, to determine whether fluconazole resistance has emerged over a decade. So this was previously done and published in 2011, and then now we wanted to repeat the study again in 2017 to see whether fluconazole resistance had emerged. So in 2007 to 2008, the geometric mean was 2.08 micrograms per mole and 4.11 micrograms per mole in 2017. The MIC50 increased from 1 microgram per mole in 2007 to 2008 to 4 micrograms per mole in 2017. The MIC90 increased from 2 micrograms per mole in 2007 to 2008 to 8 micrograms per mole in 2017. We found that in 2007 to 2008, there were 61% of participants that were female with a median age of 35 years. In 2017, 61% were male with a median age of 36 years. There were more participants on combination induction antifungal treatment. So this included fluconazole and amphotericin B, included in the 2017 survey compared to 2007 to 2008. The clinical outcome data for participants in both surveillance periods are shown in this table. We found an 11% increased unadjusted odds of death among those infected with strains with an MIC of greater than or equal to 8 micrograms per mol versus less than 8 micrograms per mol, though a 34% reduction to an 86% increase was also compatible with our data. So these findings led to new fluconazole dosing recommendations from the Southern African HIV Clinician Society for Management of Cryptococcal Meningitis. The guideline recommends a fluconazole induction dose of 1,200 milligrams per day in combination with amphotericin B, and a consolidation dose of 800 milligrams per day. According to findings from the ACTA trial, flucytosine is the best combination with amphotericin B. Since participants on amphotericin B plus flucytosine combination therapy have the lowest mortality, as seen by this lilac line in the graph. So although flucytosine is not registered in South Africa, a flucytosine access program is currently being expanded across South Africa for the treatment of cryptococcal meningitis. So just moving on to the conclusions of the study, we found that South African clinical senior-formans and C. gattii isolates showed a high genetic diversity, with the VN1 and VG4 molecular types being the most dominant. Males were more likely to be infected with a non-VN1 genotype. HIV seropositive patients had a 10 times increased odds of a VG4 infection. The VNB molecular type was not highly virulent in a Galleria melanella larval model, and the VNB strains in our study were very closely related by whole genome phylogenetic analysis. We found that some of our VNB strains were related to Brazilian strains, and some of our VG4 strains were related to Colombian VG4 strains, suggesting some level of relatedness and donation of genetic information between strains across geographical regions. Whole genome phylogenetic analysis of South African VG4 strains confirmed the high genetic diversity, with most strains not being closely related. There were 2 small clusters of closely related strains observed, with one cluster consisting of patients from a single province of South Africa, suggesting a similar environmental source. We also observed that the fluconazole MIC50 and MIC90 values were twofold higher in clinical South African senior-formans isolates collected in 2017 compared to 2007 to 2008, and our findings led to higher fluconazole dose recommendations for induction and consolidation phases in the 2019 Southern African guideline for HIV-associated cryptococcosis. And I'd just like to acknowledge the following people. Thank you.
Thank you, Serisha. Very nice and interesting talk, giving like new insights in South Africa. The talk is open for questions. If I could start with 1 or 2, which is interesting to see that in the neoformans population, you have more female than male patients. Yeah? So are there any clues why this could be?
So you're saying for the VG4s?
For the neoformans, the neoformans.
You're talking about the 2007 to 2008 versus 2017. I'm really not sure because
it seems like the numbers have reversed, you know?
Yeah, because normally it's males that are mostly infected with senior-formans, but it was, and I mean, we found more, I think, females infected with C. gattii. So I'm not really sure what this could actually mean.
And the second question was also, you see also an increase of resistance, or not an increase of the MIC values over the two periods. So does it mean that more people getting treated and so on, more people are exposed, and that's why they need higher doses, because you have a lot of like prophylactic treatment in some of the patients, or why could this be?
Yes, it is possible. Because most of these patients are HIV infected, all of them are, so they're already getting fluconazole for candidiasis. It's also possible, we speculated, that maybe the crops are getting azole pesticides. So the crypto strains in the environment are being exposed to these azoles and maybe acquiring the resistance from the environment. So yes, we can speculate either, but we didn't actually find in terms of prior fluconazole treatment being a factor for resistance. But because our data was very sparse in terms of that, so we couldn't actually conclude if that was the reason. We can only just speculate. But really, fluconazole shouldn't be used. I mean, it's easily accessible, but it's better that flucytosine should be used in combination with amphotericin B.
Okay, are there any questions from the audience for Serisha?
I have a quick question, if I might, just related to what you were talking about. Do you have any environmental samples of cryptococcus from sort of the current era to see if that change in resistance is reflected?
So from this study period, no. We haven't actually done any environmental work, but it's possibly a future study that we can think about.
Yeah, thanks. We saw probably a similar drift in susceptibility patterns for cryptococcal isolates from Vietnam over a similar period, actually. So thanks for sharing that.
I think it's a good question, Justin, about the environmental samples, because I think we need to look at the environment to see what it reflects in the human population. So I think it's probably worthwhile to go back to this, Serisha, later on, if you can. Tanya, yeah.
Thank you very much for that presentation, Serisha. I think that the issue of particularly fluconazole resistance in cryptococcus in human health is becoming increasingly relevant. And at the moment, we're actually developing some global guidelines for fungal disease, or for cryptococcosis in particular. And there is debate about whether we need to check susceptibility to the isolates regularly or not. And there are two arguments about that, which leads to a question. One of them is that the overall median MICs for both neoformans and gati are moving up, so that the distribution of MICs is moving towards MICs which are 32 and 64, but there's a very low proportion of isolates at that level, and they're, of course, relatively untreatable with fluconazole. However, in Australia, really, they've stayed around 4 to 8 and haven't changed all that much. So I guess the question is, do we move to a blanket suggestion of testing susceptibility of all isolates, or do we make it a sort of geographical recommendation that if your epidemiology is telling you that there are, say, 5% isolates with high MICs, or that there are virtually no isolates with high MICs, do we have a different clinical recommendation? What do you think? You can help me because I'm writing these.
Okay, that's a tough question. In terms of, because I know that incident episodes of cryptococcus, it's not useful during an MIC. It's rather in relapse episodes. But maybe in terms of, the thing is, if you're going to keep it to the geographical, there's pros and cons to both. If you're going to keep it to the geographical reasoning, you may miss a few, but maybe for the ones that you're going to miss, if we choose that option, for the ones that you're going to miss, maybe you can follow these patients up to see if they are responding to the treatment, if the treatment is working. So maybe go with the second option, but yeah, it's a tough one.
Okay, well, that's what I'm leaning to, so that's good. Thank you.
So your close connections, another question or comment is like, it's nice to see that what we found or other people found already before, that there's a close connection between the genotypes between South Africa and Brazil. That you found this in your genetic data too, for both the main VNB group and also for the VG4. But the idea from Anastasia was, because of the genetic diversity in Africa, it goes from Africa around the world, but it could also be the opposite to be there, because you have the same kind of genetic. So what do you think you tend more from your data, what you found? It's one or the other, or both?
It's possibly both, because now that they found, well, initially they thought the VNBs were just Southern African, and then now they found those VNBs from Brazil, and they could actually be more of these isolates, but we're just not finding them in the other continents. And so I'm thinking maybe it's just a whole mixing. Before, I mean, like Joanna Rhodes' paper in 2017, before the continents even separated, there was some mixing going on. So it's possible that it's both hypotheses. It's not one or the other, although I only said the out of Africa hypothesis in the presentation.
Yes, and there was also, you comment a little bit on the new VG5 type from Zimbabwe, so how Zambia, I think Zambia. So do you think that if we look further, that we find in other parts of Africa, or is it maybe that Zambia has a very different climatic region, that this may not be found in South Africa, and only in areas which are similar climate than Zambia? Because you couldn't find any in your study at the moment.
Yes.
And you concluded that maybe it's just environmental, hasn't reached a human infection. But even then, I think, of course, there's no clinical isolates at the moment from Zimbabwe, from Zambia itself. But do you think that if we look carefully, we found more, or is it really geographically restricted to climate conditions?
So I would think that if we perform more environmental, well, we have to do some environmental studies in Southern Africa, in the neighboring countries, then it's likely that we would find it. Because I mean, in terms of the environment being dry and subtropical, we can also find similar conditions in South Africa, for example. I haven't been to Zambia, but I would think it's not exclusive to Zambia. It could be in the environment anywhere else in Southern Africa.
Okay, any more questions from the audience?
Wieland, I've got a quick question. Thanks a lot, Serisha, for your talk. So did I understand it correctly that the VG4 molecular type is more prevalent, and you also found that it tended to have higher MIC values, but is that sort of a clinically relevant thing, or is that just because you were seeing that because the isolate is more prevalent?
So I didn't actually mention this, but we compared the MIC 50 values between VG4 and non-VG4 isolates. We found that there was one double dilution difference between them. So actually the non-VG4 isolates had a higher dilution, had a higher MIC50 compared to the VG4 isolates.
Okay.
But this was just one double dilution between them. And so in terms of if there's any MIC differences between the different molecular types, we're unable to show that with our data. We actually found that the VG4 isolates were more susceptible, but we don't know what could be the reason for this. And we also had very small numbers of the other molecular types.
Okay, Sebastian.
Yeah, thanks a lot for the chat, for the talk. My question is, have you mapped your whole genome sequencing data to your MIC values? Because if you look at other studies at other organisms, usually what you find is that there's clonality, some kind of clonality that's driving the differences in geometric means, and that, you know, once you get a replacement event or a founder effect, that's what's driving it, as opposed to really emergence of resistance, which is what you're suggesting.
Yes, so for the 2017 study, we didn't actually perform any whole genome sequencing on those isolates. So maybe a further study would be to look for those, for the relationship between MIC value and the genomic data. For the other studies, the neoformans and the Gattii studies, we didn't actually look for any of the, I know one of the examiner's comments were the aneuploidy, if we've looked for this and related back to the MIC value, but we haven't done that as yet. So maybe we can think about it as a further study.
Okay. Well, are there any more? I think there are no more questions in the chat. And I think we have no more questions online now. So I like to thank everybody, and especially Serisha, everybody for being here, and Serisha for giving the talk. And I think it was great to see a gap in epidemiology data filled by your study. And I think we're looking forward to see the follow-up study, look at the environment maybe. And we wish you all the best. Yeah? Thank you.
Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.